Hello, everyone. Welcome back to Interview with Scientist series. We are on episode 2 of our series, and our guest for today is Annu from Czech Republic. Uh, we will be talking about sepsis and electrophysiology in rodent brain. I would like to briefly introduce you to sepsis. For those who are not aware of it, sepsis is a life-threatening condition that affects millions of people worldwide and can cause multiple organ dysfunction, including sepsis-associated encephalopathy. So I'm not the subject matter expert in this field, so I'll pass the mic to Anu to explain what is sepsis and also give an introduction about herself. Over to you, Anu.
Hello, hi everybody. So I'm Anu Kala. So I'm doing my PhD. I'm in the fifth year of my PhD in the field of experimental neurophysiology, and I'm doing my PhD from Charles University. And the major focus of my study is to study the impact of sepsis on brain oscillations and hippocampal network dynamics. So we do a lot of electrophysiology in our lab in rodents, and yeah, so the main region of interest is hippocampus. So we do local field potential recordings and neuronal spike recording from hippocampus. So sepsis is a life-threatening condition that happens due to any bacterial or viral infection. So it is known that these patients in the acute phase of sepsis have severe sleeve fragmentation. So, but this is not really well understood, and also it is known that the quality of sleep actually determines the outcomes in these patients. So it's very important to study sleep during sepsis. So that's our main objective. And using hippocampus as a model system, we are also trying to understand what goes on in the memory impairment, like what goes on in the memory of these patients, because these patients are at a high risk of developing memory impairments in the later stages of their lives. So, which makes it very interesting model to study, spatial navigation, what goes wrong at the level of hippocampal spatial memory impairment. So,
Okay, that was interesting because I was about to ask what is the relation between sepsis and hippocampus, but you already
yeah.
defined it
Actually,
because,
hippocampus is one
yeah,
of the first organs that gets affected during sepsis, and central nervous system in general is one of the first organs that gets affected, but
okay,
very little is understood, even
okay.
though, yeah.
So you also mentioned local field potential, and I'm not an electrophysiology nerd, so could you explain what is that?
Okay, so local field potentials are the potentials which we record from the extracellular space in the brain. So you can imagine what we do is that we implant electrodes or bundle of electrodes, which we call tetrodes. So we record the changes in potential differences that happen. So it's like in time series, we record these changes, and they are eventually these brain oscillations that we are recording. So it's a kind of network state activity from hundreds and thousands of neurons.
Okay, that's very interesting and sounds very fascinating to do. I
It
wish I
is.
could have tried this in my PhD or postdoc. So, moving on to our next question, in what ways can rodent model of sepsis and electrophysiology inform the development of novel therapeutic strategies?
Okay, so if we talk about what happens in human sepsis, so human sepsis is a very complex, uh, has a very complex pathophysiology.
Yes.
So these rodent models are able to capture some aspects of it, if not all. So we have different types of models. For example, cecal ligation and puncture model, and endotoxemia models. So these cecal ligation and puncture models are really good at sort of mimicking the complexity of human sepsis. So most of the times, they are the gold standard. But also they have their own limitations. Like there's high variability because from animal to animal, these procedures they need to be highly standardized, but that usually doesn't happen because of, let's say, the size of puncture or the length where these punctures are made. So it has its own limitation. And also, we have these endotoxemia models, which are much more standardized, but they are suitable to capture some acute phase aspect of sepsis. So it
Okay.
really depends on your question of research, like what you're interested in. So I think these models are suitable for answering different questions. And we have seen similar outcomes, like what we see in humans. We have seen similar outcomes
Yes.
during sepsis in these animal models as well, electrophysiologically speaking. Like, for example, we have seen brain oscillation disruptions in animal models, just like humans. So, which shows that these models are actually able to capture what goes on in brain in terms of electrical activity. So, I would say, of course, they are good, and if we are somehow able to reverse these effects by using different therapeutic targets, if you are able to reverse these effects, electrophysiological effects, then and if we can somehow correlate it to the outputs or uh, behavioral outputs, then I think it would be a great model.
Okay. So
Yeah.
the other question was, do you just do electrophysiology or do you also do pathophysiology?
So I would say it's electrophysiology during pathology because
Okay.
my animal model system is a disease model system.
Yes.
So I'm doing these recordings in an LPS model of sepsis. So we
Okay.
use a very high dose of lipopolysaccharide and do
Okay.
these recordings. So,
Nice. Because LPS reminded me of my rodent models in epilepsy, toll-like receptors, which were
yes.
knocked down.
So you have the answer, right? It
Yes.
causes epileptic discharges, which are the markers of these disturbances or
Yes.
disruptions in oscillation. So in
Oscillation.
some
Yes.
sense.
Yeah. So, how does sepsis affect somatosensory evoked potentials?
Okay. So in these, like people have shown that in rat model of sepsis, in a CLP model of sepsis, they've
Yeah.
shown that the latency period is prolonged, and also these amplitudes are pretty high. And so they
Okay.
can actually be used as markers for sepsis associated encephalopathy, I would say.
Okay. So, what are some of the key similarities and differences in electrophysiological changes during sepsis
Okay.
compared to a normal or other
Yeah.
pathological condition, for example, epilepsy, as we mentioned?
Okay, so this is my favorite question, I would say, so far.
Right?
So I would actually start with, yeah, we can go to epilepsy, but I think one of the very similar things that happens in another pathological conditions is narcolepsy. So we all know that narcolepsy is a condition where we are not able to maintain sleep-wake patterns. So there are sleep attacks. So in and okay, I'd like to. I'm not publicizing my paper, but maybe it's a good platform.
Yes.
But we have recently submitted our paper, and it's yeah, the preprint is available, which you might want to see. So there we have basically tried to understand what goes on in the acute phase of sepsis in brain oscillations, and
Okay.
we have categorized these oscillations into REM and non-REM like activity. So we have seen that LPS or sepsis causes extensive fragmentation in LPS animals, yeah, a group. So we also saw that these states within themselves that they're highly unstable. So the
Hmm.
velocity, you can imagine some parameter which is describing how fast something is going. So these velocities are significantly increased in the LPS group. And one paper which shows similar things in narcolepsy. So this really fascinated me that okay, this there are some similarities. However, there is one more. We know about Parkinson's, right? So in Parkinson's, these velocity parameters are absolutely like totally opposite. So they are significantly decreased,
Hmm.
but those parameters are not manifested into sleep fragmentation or state fragmentation. So we speculate that these changes that are happening within the states are actually causing the sleep fragmentation in our model and also in narcolepsy. But the reversal of this velocity parameter is not really making these sleep patterns more stable or more consolidated in Parkinson's model,
Okay.
which means that these changes are not as direct and as straightforward as we think they are. So they are pretty, they can be pretty different.
Okay. So we have been talking about epilepsy, or we have taken that term multiple times. So is there a relation
Yes.
between sepsis and epilepsy?
Yes. So these patients in the acute phase of sepsis, polysomnographic recordings in these patients, they've shown that these patients are at a high risk of developing seizure-like activity and epilepsy.
Yeah.
And in LPS models as well, people have shown similar trend of increased uh, seizure-like activity. So it's kind of showing some imbalance in excitation to inhibition ratio of
Okay.
neuronal circuits, I would say.
Okay, that's interesting. So how can we translate this studies in rodent models to humans?
Okay, I think this is an overlapping question because I feel that in humans we have already shown that in the acute phase of sepsis there is enormous fragmentation,
Okay.
and in animal models we see similar things.
Hmm.
So we are somehow able to capture that aspect of it, and because these parameters are pretty much similar. For example, there's neuroinflammation in humans as well as in rodents. There is electrophysiological marking or differences, so which are very much similar. So I feel that using, if we talk about, let's say, I think I'm diverting quite a lot, but I feel, for example, people have shown, this is one of the studies, very interesting one.
Yeah.
People have shown that injecting interleukin 1 beta in brain directly into the brain causes similar state fragmentation, and we
Okay.
know that there is extensive cytokine storm in humans as well during sepsis. So
Okay.
which, it's not a direct mechanism, I'm not saying, but this is an indirect link because so far, to the best of my knowledge, I have not
Yeah.
come across any study, any interventional study that reverses these effects,
Okay.
electrophysiological effects.
Okay. Interesting. So what are the next steps in your research?
Okay, so this is a very
Maybe
interesting one again.
confidential, I know how research is,
Uh,
but
I'd like to keep it, but it's okay. I can probably share because nobody has so far touched this area. I
okay.
don't think anybody is going to do it overnight. But yeah, so what we think is that, in sepsis patients later in their lives, they are at a higher risk of developing memory impairments.
Yes.
But a direct link of sleep disturbances and memory impairment hasn't been shown,
Okay.
or the mechanism of memory impairment in these patients has not been shown at neuronal circuit level.
Okay.
So we are trying to use hippocampus as a model system because we know hippocampus is great for spatial navigation, for memory processing, memory consolidation. So
Yes.
it has place cells, right? Very specific place specific cells that fire at a particular location. So we use this as a template to study what goes wrong in the processing of these maps in the sleep states, because
Okay.
sleep is fragmented in these animals. So that's our main agenda.
Okay. Sounds interesting, and I'm not going to scoop it because I'm not in research anymore.
Yeah, I hope
So
so.
no one will do that, and it's not a overnight thing to do. Developing an
Yeah,
animal model takes time. So,
yeah.
we have talked a lot about your research, but we had some guests who are here or who might who will see the recording. They were interested in knowing about the process of obtaining a PhD, or how did you get your PhD position in Czech Republic or in Europe? So, if you could tell about that.
Yeah, sure. So I was in India that time, and one of my friends, one of like somebody I knew, so that person like recommended me this lab because his friend had worked with this team earlier. So I went through the like advertisement, and I directly emailed the professor.
Okay,
So I actually got a reply from him after 4 to 5 months. So it really depends on when they have the position
funding.
available. Yeah, funding
Yeah.
and position. So it was pretty random for me. And once he like replied, and we had an informal interview sort of thing in the initially, and then he
Yes.
asked me to apply through a regular portal. So that time I went through a formal sort of interview with them, with a team of 5 to 6 people, and it was pretty good. Like, people here are really warm, kind, and
Okay.
they're really nice. So it wasn't that uh, difficult, I would say.
So, how was the transition from doing a master's, I guess, in India to a PhD in Europe?
Yeah, so like masters in India, we know, right? It's a very different culture. So of course there was a great deal of cultural shock, and I mean it was very new. But here I feel it's much better in terms of funding, in terms of, I would say, social interaction with people. Like here we are valued more as people or as
Okay.
PhD students. I mean, this is my personal opinion.
Yeah.
uh, I feel we are valued a lot more here than back in India.
Back in India. Yeah, I did my PhD in 2015 from Germany, started my PhD. So it
Yeah,
has been, I know things might have changed from 2015 to when you started.
yeah. I
So,
started in 2018 actually.
so
Uh,
yeah, that time, yeah, I moved out of Europe. S-
okay,
So what are some career plans for you after your PhD?
okay. So actually, I recently got my postdoc in Berlin at
Congratulations.
Charité. Thank you.
Okay.
So I'll be working with Dr. Schmitz. So I'll be
The
moving next.
advancements.
Yes, yes, the Schmitz. Yes,
He was
you
one
know
of
him?
my, yeah, he was one of my, like, I collaborated with him.
Okay, nice.
I also
That's
worked in
great.
charity for like 6 to 8 months, something like
Okay,
that.
nice. How was
So
your experience?
it was good. I was working with Imre Vida, Dr. Imre Vida,
Okay, I have no
and
idea. Sorry.
they collaborate with each other, Imre and Dietmar Smith. So you
Okay.
will see that.
Yeah.
That's nice. So, but you always wanted to do a postdoc, or you wanted to move away from academics, or you have future plans of moving away?
Okay. Actually, I never really thought about it deeply, but like I'm taking each day, I mean each step as it comes. So
Yes.
for me, for example, I wasn't sure. Like when I started off with my PhD, I wasn't sure if I'll stay in academia or research. So I was like, okay, let's give it a try. And with time, I think I developed this interest, and it's just keeping me like I just want to be in research for now, but I cannot say like 5 years down the lane if I'll
Yeah.
move to industry. So no plans, no
Okay.
future plans, but just living in the present.
No, that's important, living in the present. But yeah, I was, I think I mentioned this multiple times, but yeah, I was a scientist at University of Toronto. And then afterwards, I moved out of academics to industrial positions. Now I'm a digital marketing specialist, which is
Oh,
completely different from
fantastic.
neuroscientist, from being a neuroscientist. So I'm a mix of both now, a crossbreed, you might say.
That's great.
So yeah, and I would open the podium for our guests to ask any
Yeah,
questions if
sure.
they have any.
Yeah, sure.
Siddharth, Parvati, and Vivek, please feel free to ask your questions by unmuting yourself.
I had a really nice talk here. My name is Siddharth, and I actually wanted to ask you what was your inspiration to take this as a career?
Okay, so that that sounds like an easy question, but I feel it's a process. Like you don't really uh think about things, like you don't really think too far in future. So for me, it was a regular thing. Like okay, you did a bachelor's and then master's, and I remember in my master's, during my master's thesis dissertation. So while doing this summer no it's the dissertation sort of thing, so we have to be in a lab for 6 months and do all the lab work and be with PhD students and accompany them for everything. So I wouldn't say that was the time when I decided, but it was by the end of my master's that I started to, like, if I got some results that just made my day. And I think till date that is what has been happening. Like it's I cannot explain why, but it's just weird. I wasn't prepared for this question.
The
Sorry.
jackpot question.
And results take us forward much more. So it's a really good inspiration to have. And thank you for your talk.
Actually,
Um, Really
actually,
nice.
I have an answer. I have an answer. So
Go for
when I was training somewhere in Isa Mohali actually. So there was a professor there who told us that, hey, I want to inspire you guys. So for that, she was like, that just think about it, that there's something in the world which only you have uncovered, like nobody else. So that is your gem. And then she was like, so you people are precious, you people are special because you, for the first time, you discovered something that the world just did not know about. And these small things, they just
Yeah,
inspired me.
that's really nice. So that, yeah.
Yeah.
Yeah, thank you so much. It was really
Thank
inspiring.
you.
Any more questions from our guest?
Hi, Annu. Parvati this side
Hi.
from India.
Hi,
I
Parvati. How are you?
Uh, Very inspiring talk. As someone who is looking for PhD positions in the Europe, I'm very curious to know about how the work-life balance is there compared to back in India. Like, why? How would you comment on it?
Okay, so I would say it's purely subjective. Because, but the good part is that you have the choice. You have a choice to make, like you can either have it or not have it, which is a luxury. In India, you don't have a choice. So here, uh, you can definitely, on weekends, you're not expected to work. I mean, you are no, not really. You're not really expected to work. It's for you. Like if you feel motivated enough, or you are just going crazy, you won't sleep if you don't see a particular result in this particular time. So then it's something, but other than that, I think you are good to go. I mean, you can definitely have time off, you have holidays. There's no concept of holidays in India for PhD students. Like they do go once a year, but that's something which you have to really ask, or it's not something that you say that it's your right or something. So that's, and this is personally, like this is only my opinion, I'm not generalizing anything, but I think you'll have a good work life balance here.
I kind of agree with you. Unless you are doing really hardcore time limited experiment or time oriented experiments,
Yeah,
like having a mouse meeting or anything related to that or cell culture, you will get your time off. Right, Saturdays, Sundays are for you. Then you're
yeah,
not expected to work unless you get a supervisor who wants you to be there. That's a different case,
exactly.
but most of them are not like that.
Yeah. And if you have your time limitations or time bound experiments, I think that's okay. And they can be compensated for some other time. Like, for example, you can probably take some, if you have a good understanding with your supervisor, you can actually take a day off in the weekday, I think.
Yeah,
Because our supervisor is really cool and nice, and he's
yeah.
really nice. So.
And I have done the same. Like, okay, I worked over the weekend, Saturday and Sunday, because of some experiments. So he said, you can take any time, or you can accumulate these and take those extra time off whenever you
Yeah.
want to. So, yeah.
Thank you so much. Thank you for the reply. Thank you.
And thank you for keeping yourself awake at like around 2 o'clock in
1:30. Yeah.
India. Any more questions? We still have some time left. Okay, I don't think there are any more questions, but it was really a pleasure talking to you, Anu, and especially about sepsis, because this was something new to me. used to the term epilepsy. But yeah, I can see how sepsis and epilepsy are connected. And if I was in research, what I could have done next. Maybe there was
Nice.
a collaboration step here.
Yeah,
But
I
yes,
sense collaboration.
yes, collaboration is the key right now. And it was also way back. And it's the future because once you collaborate, you can get more ideas and spread your wings, as I say.
Correct. Correct.
Thank you very much for your time. And this recording will be available on our YouTube channel.
Okay, thank you so much. Thank you so much for having me.